Good evening. This is Sue from New Jersey Mycological Association, and tonight it's Taxonomy Tuesday, where our members share their recent fungal finds, or lichens, or slime molds. If you have something and you've joined us, if you put your name in a chat, we can call you in order if you haven't already sent in an email. Would anybody like to start?
Sue, you should start.
Oh, okay. I told you ours is pretty boring, but it is, I guess, beginner mushrooms, the ones I have found today. Um, let me s- Well, just like last year, I have jack-o'-lanterns coming up now in our local park.
What? That's crazy.
It happened same thing last year in June. I think it was more like the end of June last year, but now it was this week they came up.
Oh my gosh, how interesting.
I did check on the iNaturalist, and it's not unusual because it happened in their, occurrence screen or whatever was sort of at the bottom of the parabola of when it will appear. This is obviously they're clustered, they're decurrent, and of course they're poisonous. But it's always by the same spot where I have to walk because they're not allowed to park anywhere near it. That's part of the park.
That's a good clump there.
Yep, and that's only part of it. There's more. I figured that was enough to show that it was quite a few of them. That's why I can see it right by the road, but I just can't park anywhere near it. Next door to me, we have Chicken of the Woods coming up. This is the white-pored one. It still doesn't look much of it, but I can see the little orange coming in there, so I know what it's going to probably be.
That's a beauty.
And the last one, we can go through a discussion of levis versus Trianus. This is not very good pictures on my part. I found one specimen in a place where I'm going to be walking with a group on Saturday. I wanted to check out if they had any fungi at all. They had some uh, Megacalibia romanii, but this is the best find I found, which was just one of this Pleurotus sitting on a piece of white wood. And you can see everything is very yellowish, but it's not really as that yellow as that. It's obviously a white rotter because it was sitting on nothing but cellulose just right next to a stream. Uh, Again, not very good pictures, but it's very hairy on the stipe and it is getting old, and that's why it's turning yellow. Okay, and that's all I have.
Is there like a one of the ends of that log? Is it like in the water?
No, it's not. It was parallel to the path.
Hmm.
And it was also a very small piece. It was only maybe 6 inches wide and pure cellulose, nothing that even tell me what it is. These woods are pretty bad. They're almost all maple, um, sweet gum, and tulip poplar, no oaks. Just by the nature center are there more diverse trees because it was a home that they converted into a nature center, and they have things like Atlantic cedar planted there, and bamboo, and other strange things that you wouldn't expect to be found there. But there was really nothing. This was the best find. Again, I don't know where this is supposed to be found, but I think the diagram of seasonality for this also still fits of when it can be found. Okay. And I don't, not quite sure how I get to the seasonality thing from the desktop version. There you go here. Just seasonality. Here it is. So here we are. We're at the beginning, just like the jack-o'-lantern. We're sort of at the beginning of when it can happen, and we won't peak until August.
Mm.
And see, these is pretty much like what I found. It was on the ground. The only other times I have seen this species was on a standing dead tree. Nothing this basically that close to the ground.
Those are on wood, aren't they?
Yeah, they're on wood, but yeah, this one again looks like it's on the ground. It doesn't look like it's on a vertical, coming out of a vertical tree.
Right. A fallen, like,
Yeah,
yeah. Okay.
yeah, it's coming up as opposed to coming out from a tree.
Yeah. That's how I've seen them too, I think. And I don't see that much, but interesting.
Uh, would you like to go next, Hunter?
Sure, I could. Yes. And I can do a screen share.
Okay.
Hopefully, let's see. Oh, do you see anything?
It's coming.
Okay. Okay. Uh, okay. Do you see INAT?
I do now.
Great. Okay. So this is like incredibly boring. Trichaptum biforme. But it was pretty, and I took a photo, and I was hoping to find those little pinheads, because I thought they are like on every single one, but I don't have them. They're not here. Um, I did do microscopy, and these are the spores I uh, found, and they came out pretty quick, so that was nice. I didn't have to wait forever, um, and so that's that. And then,
The
uh,
um, the Phaeoclisium, while it is common, it's not nearly on every cap. It's you know you could look at all the Trichaptum biforme in the woods and only find a couple, in any particular set of woods, or only a couple fruitings.
oh,
And
okay,
Trichaptum biforme is so common. You know what I mean?
yes.
Yeah, that's my experience also. It's a low percentage of times that you find the fairy pins.
Oh, okay. So I should have looked harder at more specimens.
And
Oh,
they are, you know, when we look at them here, they don't seem as small as they really are. You can barely, if you know what they look like, you could make them out with the naked eye. But the first time I saw them, someone pointed them out to me, and I'm looking at the thing like I don't see anything.
oh,
So
no, I expected not to see anything, and I brought it home and I looked at it under my whatever it is, like this. Is that a spider? Look at that spider.
oh yeah,
Wow. Oh my gosh. It's got an egg or something.
yeah, a little egg sac.
Yeah. Holy moly. Um,
But even this photo, for example, like this photo is good enough that if they were there, you would be able to see it in this photo. You would be able to see, even if they were out of focus, you'd be able to tell, like, oh, there's probably Phaeoclisium on that.
right. Would it be on which side would it be on? I think I was
Cap,
looking at the wrong side.
cap.
Oh my gosh. It'd be on the That's a riot. Maybe it is here. So this is the side. I was looking at the other side for it.
Yep, it'd be on the top.
Oh my gosh. Okay, but it looks like it indeed is not there, right?
Right.
Okay.
Yeah, I would agree with that.
Oh, that's
But interestingly, you can see this one geo-orienting,
I
right?
was just
So
gonna say,
yeah, you can see the new growth is growing at a different uh what angle, just
yes.
trying to shoot those spores down.
Mm-hmm. Yeah. Interesting.
Yeah,
Okay.
the tree must have fallen down when it already had the Trichaptum on it.
Yeah. Okay, cool. Okay, and then this is a new genus name for me. So, what I saw in the woods was a lot of first just for the season, and I probably already have seen them, but like I noted them for the first time this season. So that's all I've got is stuff like that. But I thought this was still dic, duc, Ductifera pululahuanum, but I believe this is what it's called now. Are you all familiar with this new genus?
Igor.
Yes, this name is incorrect. Well, maybe not this name. The application of this name is incorrect in this country.
They keep changing my
It's called
occurrences on iNaturalist to this new genus.
it's called Protohydnum, but it's not going to be pululahuanum. Pululahuanum is only found in South America. We don't have it here in North America.
I know the name is
So it's album, Protohydnum album, I think the correct name is.
Oh, okay. Okay, great. Mm-hmm.
Well, that's easier than the old name.
Yeah, Ductifera pululahuanum. Yeah.
Pululahuana, yeah, it's a local name from South America. It refers to
Well,
either
where
a geographical location or an Indian tribe. I forget exactly.
I believe it's a town in Ecuador where it was first discovered.
Okay.
That's my understanding of it. But yeah, and I did the microscopy too, and things were very tiny with this, but I was able to see the sausage-shaped spores.
Sausage.
And then I don't have a good picture, but I saw a lot of clamp connections on the hyphae. And then what else did I see? I feel like I saw something else, but maybe not. Maybe not. But yeah, it was interesting. But these were really tiny stuff. I also found my first bullet of the season, and unfortunately, I believe it's a Tylopilus felleus. It did bruise brown. It does have reticulation on the stipe, but and I mean it kind of looks white near the top, but I it's really more brown reticulation.
What kind of trees
It
was
was
this?
it was growing on a very degraded white pine stump. Like you could
Oh, okay.
barely tell it was a white pine stump anymore, but I'm sure it was because it was in a white pine forest.
Yeah, that sounds like a Phaleus
Yeah, it's
habitat.
quite
It's just a
typical.
strange looking one.
And
And
I put iron salts on the that's that orange there. This was a pretty quick reaction. It wasn't like a flash, but I didn't wait around too long. So that orange came from the iron salts. And then here's a close up of the pores. And then I did the spores. I believe these are the spores of that mushroom. And I hope. And so yeah, so that was cool. And then I know we've seen this before. Everything I'm gonna show you, we've talked about before and seen before. But I believe this is Dacrymyces capitatus. It's really beautiful, just so pretty. But these are so tiny. But we are lucky we can blow everything up. So some of them look like little tiny cups, but but most of them, the center of the cup was convex. And I think people call that like a pincushion look. So like you see here, but the very youngest ones, it was more concave. And then they have a little stem. This is actually upside down with this other microscope thing I have. It's not really a microscope, it's like a magnifier thing. But I wanted to show the little stem. Uh-oh. Sorry about that. You still there, everyone?
We're still here.
Okay.
Yeah, your share is here, but we lost your mushroom picture.
Okay, hold on. Oh my gosh, stop calling me, whoever you are.
I was gonna say those were really good photos, though.
Thank you. I worked really hard to try to, to, get it. And I did microscopy. And I know, Brandon, I think you'd done it. And it's very easy on this tiny thing. I mean, it was really easy. And I was able to get the a tuning fork shaped basidia. And
Nice.
yeah, and there's another one. And also the spores, which are pretty cool. I think Quo said they're, what was the word he used?
Septate.
Tardily septate, which I don't exactly know what I mean. So I think they have like a lot of oil droplets, but then are tardily septate. So it must be like, what does that mean? Like after a length of time, they turn that way? I don't know what that means, but these were the spores, and oh, here's
Yeah, I don't know what that means either.
Maybe the spores kind of look like humpbacked a little bit, so maybe that has to do with like the septations are like smaller on one side and larger on the other, and that's what makes it have that little arc.
Oh yeah, maybe. Yeah, that could be. I don't know. I should probably just like look the word up. I know what tardy means, but tardily, I don't know. Um, so yeah, so that's how I got really. Oh, but that's not exactly true. I can show one more thing. Um, I I ate Lyophyllum this week, and can you see it?
Yes.
Okay.
And you're still here.
What's that?
And you're still here.
And I'm still here.
Yeah, you ate the biforme.
Oh,
You're still here.
still here. I know. And I'm still here. But I have to say, I don't think I felt that great after. Like I didn't have any, I didn't have any like vomiting or diarrhea or anything, but I was a little nauseous
That's psychosomatic.
and maybe the texture was really like that's a little raccoon, really what is the word, like collagenous. It was, I mean, I think in some ways maybe that's part of why it can taste like fried chicken, because when you get that like crispy fried chicken, it's sort of like cartilagey a little bit. Like I don't know.
Do you mean it was rubbery?
Almost rubbery. Yeah,
Uh-huh.
it was work, like not a lot of work to chew, but it was work to chew. And I mean, not work, but just not, it wasn't delicate at all, but
Where did we land on the common name on this? Was it the fried chicken or was it the chicken fat
it was
mushroom?
fried
All
chicken.
right.
Fried. Gotcha.
Yeah. Yeah. I think chicken fat's Suillus American or Americanus. I'm remembering I do have one more observation to share if that's okay.
Well, now you have to sequence this one to find out what exactly you ate.
Oh. Oh. Yeah.
Now that you're still alive, you have to do this.
Oh, yeah, that's right. Now, maybe someone else wants to go. The other one's on Mushroom Observer. So if someone else wants to go, we can come back to Mushroom Observer observations later if there's time. Or it should be here. Wait, one sec. I'm here.
Found out I looked up tardily.
Oh,
It is, it's at a slow pace or late. And
Right.
yeah.
So Hunter was probably right there, like later mature spores, right?
Yeah,
Which
that's
can make you septate.
yeah. Okay, that observation somehow didn't get registered, but I can show you right here. This is okay, just making sure I would not muted. So sorry. Oh, so this I believe is Lentinus. It's a train wrecker.
Lentinus,
Neolentinus,
Neolentinus lepideus.
lepideus, yes.
Yes. Yeah. And I thought this was a pretty cool specimen. It was growing at the base of a very, very old, like very decayed white pine. There was no bark left on it at all. And so even the presentation was a little bit different than I've found. But I'm pretty sure that's what it is. Oh, and isn't there, the gills are kind of,
They are serrated.
Yes.
Jagged, yeah.
Yeah, jagged serrated gills. And then these are very rubbery. These are edible too, but I didn't do that. These
I thought you would eat them too after you ate the Lyophyllum.
no, maybe next time. I like to do one at I'm really slow go on it. But I did the spore print, it was white, and I also did the spores, but somehow this observation just didn't get filed. So that's it now. Thank you so much for letting me share.
Thank you, Hunter. Brandon, would you like to go next?
Yeah. Let's see if I can figure this out. Can you guys see my screen?
No.
Not yet.
No.
Yeah, sorry.
Are you still there, Brandon?
Are you on
Yeah.
the iPad or what are you on?
I'm on my phone. I knew how to do this, but they seem to have. There's no actual share screen option.
Uh-oh.
Oh, I found it.
Yeah.
All right. Can you see my screen?
I can.
Yeah.
All right. You see iNaturalist?
Yes.
All right. And can you see that photo decently enough? Or is it?
Yeah, it looks good.
Yes.
Okay.
Cups with some fluff on the edge.
Yeah. So this is a species of Lachnum. that I found last week, and they're very, very small. And you can see they have a little stipe, a little stem. They're about as tall, maybe if not a little taller than they are wide. And yeah, you can see those marginal hairs are quite interesting. Those hairs actually they run down this stipe.
Yeah, let's call them bearded cups.
Bearded cups, I like that.
Oh, wow.
That's them rehydrated on the back of a matchbook cover to give you a bit of scale. Here's another shot under the stereoscope, trying to highlight the maybe you can see it up here. Even the stipe has that beard.
Interesting.
So this is in inches and not very useful. Can you convert this to millimeters?
Those
Maybe
are
like
millimeters.
5 or less?
Well, each integer is a millimeter here, right?
Is that a centimeter? That's an inch.
There's
That's
both
right.
meters and inches.
I think it's
It's a
inches, right?
standard tape measure. Those increments are in sixteenths.
A 16th of an inch.
Right.
So let me
That's
do the math here.
yeah. You or Dave, that's your department. One of the
Yeah.
math guys.
So the biggest
Please
one there is
Google
about
it.
16th.
So each division is then 1.6 millimeters.
Okay. So I need to revise that because I was calling these about 1.5 millimeters each, and they're probably about the largest one is probably more like 2 or 2 plus, maybe.
So what was it growing on? What are they growing on?
So it's growing on, we'll skip right to that. Decorticated, I was guessing birch just because I found white birch, like an old uh, white birch bark. You know, everything else had decayed, but there was white birch bark all around this thing. But who knows? It didn't have any. This particular little log, stick, whatever you want to call it, didn't have any bark.
Wow.
Wood. And this is the in situ photo, which is obviously not very helpful, but I wanted to take this to show what you can do with xeric cup fungi or any fungus that has a xeric state, which means that when it experiences periods of drought, it just kind of expels all its water and goes dormant. And then once it gets some more hydration going, it can pretty much come back to life. And in that dormant stage, I'm pretty sure the cells are more or less dead, and they can kind of rejuvenate with the hydration, which is the whole, which is what xeric means, I think.
So you rehydrated that?
Yes. So this is how I rehydrate it. That's a paper towel that I pretty much soaked in water. I left a little, uh, maybe a quarter centimeter of water on top of a high folded paper towel a bunch of times so that that paper towel would retain a bunch of moisture. And then the bark would also retain a bunch of moisture because I collected, or not the bark, the wood. But yeah, this is after like 5 minutes. And then after like an hour, they're back, you know.
Wow.
Yeah, and that's when I took this photo. So that's about after an hour of rehydration. They're just kind of back, and you can make out the stipes and everything.
Neat.
So
And
yeah, just a practice. If you guys ever find like old, kind of decrepit looking cup fungi, they can be brought back.
you could tell they were cup fungi before.
Only because I'm familiar with this genus.
I
Um, Yeah, and I was like, that's probably a Lachnum. They I didn't include that photo probably, but even in the in situ photo, maybe you can kind of make out like this one here, super out of focus because when they're uh, dehydrated, they are even smaller. But you can kind of see the appearance of
Yeah, you can. And the stems, the little
yeah.
stems.
And look over here, you can see that yeah, that's probably a little cup.
That's really neat.
But then I did the micro on it as well, and the micro on these are pretty interesting. Um, I'm of the opinion that the genus is just it's one of those genera that's just in flux right now. There's a bunch of names getting thrown out. I put Virginianum on this one. There's a Virginium, there's a subvirgineum, and then all the other ones are pretty similar. I don't know.
What are those? What are we looking at?
So these are the spores here in the center of the reticle. Can you guys see that? I think I lost you for a second.
Yeah. And what shape are those spores? What do we call that? They're like tapered at both ends or
More tapered on one end,
one,
which
like a
that's a good,
cigar?
yeah, like a cigar. Yeah. There is a better word for that. I don't know. Igor, Dave, what would you describe that shape as? And they're pretty small. This is at my 430X, so, you know, eh,
Um, bacilliform.
Yeah, I think that's the word.
Or cylindric,
Yeah, bacilliform usually implies
rounded ends.
over Q over 3.
where does fusiform come into play there?
That's the shape
Tapered
of the spore.
ends.
Oh, that's it. That's the
Yeah,
yeah. Oh,
Yeah,
but it's
fusiform is more of like a silhouette, an outline, where cylindric refers, I think, more to the Q ratios.
right,
Yeah,
right.
um,
So something,
cylindric is they're just sort of straight. And they're not really highly tapered,
yeah, isn't
and
bacilliform like rod-like?
yeah, they're rounded on the ends.
Yeah, so bolete spores can be bacilliform and a fusoid at the same time.
like those
Yeah,
Tylopilus I showed, right? Kind of.
fusoid or fusiform are tapered on at least one end.
Yeah, and these terms can be helpful in descriptions, but at the same time, if there's not a picture of the thing, a lot of the times there's dashes going on, right? So it'll be like bacilliform fusoid or
Yeah,
something,
Yeah,
yeah.
fusiform means spindle shaped.
That's right. That's right. And I maybe that applies here to some of them. It's not a great photo. Because I wanted in this photo in particular, there's a lot of different structures. So we have the spores there, just right near the the reticle, whatever it's called. Here in the upper right, you can see some paraphysis. That pointed thing is a paraphysa, paraphys. And then you have an ascus that you can see the spores. Now, none of these are really good photos of those things, but you can kind of get the idea of the different shapes all in one photo. So, but there's another shape. Here's a better shot of the ascus and the kind of lanceolate. Th- well, that one's not very lanceolate, but I happen to think there's a lot of variability. So these paraphyses are much more lanceolate, and that word I have a pretty good understanding of. Right? It looks like a lance, looks like a snakehead, I guess. Um, and yeah, these two fit that bill pretty well.
Yeah, very cool.
But then the other thing when you're reading about these, um, so notice these kind of subcapitate things, um, and this is along the margin. So maybe you can guess that the hairs or the beard on this thing, they actually are subcapitate. That's what all the descriptions say. And there is variability between, whether or not these have a little, you know, head capitate, um, or not. Some of the descriptions say they're swollen, some say subcapitate capitate, but there's absolutely some swelling at the ends of these things. And believe it or not, those are the hairs or the beard that Igor was talking about.
Neat.
Yeah. All right, so that's that one. And I just have one more I wanted to share. This is a slime mold. So this is a Myxomycete and Stemonitidaceae. The chocolate tube slime molds. And I did some micro on these for the first time since taking that Myxomycete course with Dr. Stevenson. And I was excited to look at these things because I learned a lot about what you have to look at to identify these things. And yeah, one is the spores at 1000x, which I don't do good work at 1000x. So this is at 430x. There's probably some ornamentation that I'm missing there because of my level of magnification. A lot of these have either reticulate or warded at the very least spores. But in the lower left, you can kind of see one of the things that you really need to look at, or not really need to look at, but one of the ways that you can differentiate between Stemonitidaceae, and that is kind of unique to Stemonitidaceae, which is this outer netting. So if we go back here, the brown part that's sitting on a little foot, the brown is because the spores are brown. So each one of these fruiting bodies is pretty much a net holding in the spores, and you need to blow the spores out in order to identify the thing. So if anyone's ever handled these, the spores come out pretty easily. And you're left with this kind of netted structure and the shape and the size and pretty much that shape size of the netting there can give you some good information. That last photo was at 430. This one is at 100x, just to kind of give you a better idea that netting engulfs that entire central columellum, which is another thing that is diagnostic of Stemonitales, maybe Stemonitis proper. I have to look at it. I just did this micro today. Yeah, another look at the netting, and it really is. It looks like like a fish net, right?
Yeah, that's neat.
And then this is under the stereoscope, just to kind of give you a zoomed out idea of what we're looking at. So that stem, that foot that you see, extends all the way up through the fruiting body, and once it hits the sterile part of the fruiting body, the part that has the net that we were just talking about, that contains all the spores. Once it hits that point, it's now a columellum, and that is diagnostic as well. How far that columellum goes towards the apex of the thing. This species, it went all the way to the top. Sometimes that columellum isn't present. Sometimes it only goes halfway.
So you blew
Yeah.
spores out before doing the microscopy on these last two pictures?
Yes.
That's
I just went and looked at an old one of these that I have, and now like it definitely does make more sense. I
What
didn't, I didn't
to
know
blow
that
up?
that center part was like a diagnostic feature at all.
Yeah, that and the fact that these are grown in clumps. Um, it's easier to know what to look for than to actually like ID the thing. But I do know that it growing in clumps like this is also a diagnostic feature.
As
So
opposed to like because I've seen them in like almost like neater rows.
yeah, yeah. Or some of them, you'll see maybe four or five all out of one pseudothallus, which I didn't do a good job of documenting, I don't think. And the pseudothallus is like there's a layer that all of these things are coming out of, more or less. So I didn't do a good job of documenting that. But yeah, some chocolate tubes, super, super common. But I finally got to do some micro and look at that net, which, yeah, it was pretty cool under the scope. So
How'd you do that? Just because you really like, did you bother putting a cover slip on it?
yes, yeah, yeah. I grabbed like two or three individual fruiting bodies, uh, arranged them on the scope, uh, facing the same way, so I knew which part of each fruiting body I was at, depending on which way I was going. But yeah, I did the full smash mount, more or less. I know a lot of people are just getting away with looking at things under better microscopes without cover slips, but I don't have that luxury.
It feels like risky business.
yeah, I agree.
Well, cool. Thanks, guys.
Mhm.
Especially with spores, you can just like blow out.
Does anybody else have anything to show? I know uh, Dave has something, but it's not quite 8 o'clock yet. Mhm.
I could show an old uh Stemonitis.
Okay.
Then yeah, there's the netting.
Not seeing it yet.
Oh, you can't see it?
No. Did you share your screen?
Yeah, I hit the button. Share screen. Oh, I see. It didn't do the countdown.
Oh, a little stick. Convenient.
Yeah,
Wow.
it was actually what do you like the center part of a leaf that like got chopped up in the mower and was just in the lawn, and I pretty much there was some sitting like it was just kind of around this stump. And I guess that's what you were talking about, the pseudothallus. Is that it? Like around the bottom of that thing?
Yep, yep. See how they're all kind of just coming out of that black pseudothallus?
Cool. I don't know. It's weird. As soon as you started talking about it, I just see all the pictures differently. But that was a little chunk that I could pick up easily enough. And I didn't even think this was so long ago. I didn't even think to get the whole thing. I was just trying to look around at stuff, and that's the very top of one.
Yeah, nice.
Wow, cool.
And you could even see like uh the spores hanging on to bits of it in there. Which these seem
Neat.
more like a different shape, the spores of these, than the one that you had.
Yeah, for sure. They also seem bigger and more slender, which,
Yeah,
when we look at these things under the scope, sometimes we don't just take that into consideration. Like, oh, this one's reported no bigger than, a centimeter or 2 centimeters. And some of these things can be pretty large in Stemonitis proper.
yeah. Now I'm all excited to find another one now, so I could do the right kind of micro. So thank you, Brandon.
Yeah, no problem. Just another slime mold convert.
Chris, Dave, are you able to go now?
Yeah, I guess I could. Is Jeopardy over?
No, Final Jeopardy.
It's just Final Jeopardy. They're gonna play the song though. Um,
You
yeah,
don't
go
have to
ahead.
sing along. Let
Well,
me bring up.
we thought they took our. There's the music.
Yeah, but that wasn't true.
Okay. Oh, wasn't true. Oh, that wasn't the
No, it
problem.
wasn't.
Okay. Okay. Well, we turned the music off anyway. Okay, so um, what do we have here? I found stuff on Saturday, I guess. So there was a lot of Galerina paludosa in the sphagnum moss, um, in a swamp on top of a mountain, Ricketts Glen. So I think this is just Galerina paludosa, but it's really pale. Look at how pale the gills are. And I didn't save this one. It was like the one that I didn't save. So, but look at the that's velar remnants on the stalk. It's a partial veil. It gets spread out on the stalk. I think that's all this is. Um, I mean, if anybody has any experience with that species, and
Well, I was just going to ask you because I know you do have experience with that
yeah, I
species,
find a
but
lot of these, and they're
the
the
difference
gills are so
between
pale.
I mean, Hypnorum, you can tell morphologically, right? Like this doesn't look anything like what you would call Galerina hypnorum, right?
I think that's that really tiny thing that's got like deep striations. Um, yeah,
Yeah, yeah, just one of these Galerina that you find in the swamp, also.
yeah. There's a few of them. There's a few of them, but this seems to be the most common one around here, especially in the spring.
Hmm.
This paludosa. I think that's what this is. But I just thought it was interesting that it was so pale. Now, this photograph may have been taken in direct sunlight, and I may have edited it. It doesn't really look like it though.
And because I'm really like I know of Paludosa just because I've compared a bunch of things to Paludosa before.
Yeah.
Um, but when you say it's pale, it's normally more orange or more
Well, the
brown.
gills don't show any pigmentation hardly at all. I mean, maybe a tiny, tiny bit of brownishness toward the margin, but usually the gills are yellowish
Mm,
on pretty much all of these small Galerinas.
yeah. Okay. Cool. Thank you.
Yeah, so that's Otherwise, this looks like paludosa. Maybe the cap's a little lighter than usual. But anyway, that I just thought it was interesting that one kind of got me confused. So this really looks like Ganoderma tsuge, right? A whole bunch of them growing on an old tree. Except this tree is maple. It's red maple. And there was another red maple that also had varnished Ganoderma on it, just one fruit body. And these really do look like tsuge. I mean, Curticii is usually more yellow. So I posted these, and I think I said I think they're Curticii, or they might be Curticii because they're growing on hardwood. But I'm really wondering about this, and I sampled. I put some in the one of the DNA plates. I used the toothpick to sample from the fertile surface. Now, hopefully, the fertile surface is going to be immature here, so I hope that doesn't really matter.
Could we be dealing with what is it? Sessile, Ganodermal sessile. That's
Nah, I
not
don't think this is
That's
sessile.
not sessile.
I don't think so. Sessile
Yeah.
doesn't have the stalk like
Right.
attachment.
There are
It's
a lot
possible.
of people growing all different cultivars all
Yeah,
over the
that's
place.
right. That's true. That's true. Who knows what this is? And the funny thing was, this swamp where these maple trees were, where this was growing on, had have a lot of hemlocks in them. And I'm trying to remember, did we see any hemlock reishi, growing on hemlock? I don't think we did in this location. This was last Saturday, I'm pretty sure. Yeah. Wait, it's Tuesday. Yeah. Yeah. So I think this might just be Entoloma strictius, but it's kind of slender and small, and it has kind of long striations on the cap. So I sampled it. It's probably just going to end up being strictius though. But it looks different somehow, I don't know. We'll find out. The spores look like strictius. They're five-sided. They're mostly five-sided, and maximum length is a little over 12 microns, so that fits strictius pretty well.
Yeah, I've no opinion other than it looks a little off, right?
Looks a little off for strictius. So I sampled it for sequencing. But I know they get sick and tired of sequencing strictius because I guess a lot of people send them in because they're pretty common and they're big.
Yeah, and I was just gonna say maybe that supports what why it's reported as so highly variable, right? If it's been sequenced so much, and that
Oh, yeah.
still seems pretty true that
Uh-huh.
it's just a
Okay.
highly variable thing. But
Well, maybe I'll find out about, at least in this case. So I got more Mitrula today. I picked a whole bunch of Mitrula in a spot, not a different spot than where these are from. This was just one in this sphagnum swamp, just one Mitrula that was growing. And so the interesting thing here is, okay, so it's got the long cylindrical spores that are more or less cigar shaped. Some of them are a little bit bent. Some of them are, a little bit bent, maybe a little bit tapered on the ends, but they're not crescent moon shaped. They're not boat shaped. So, but the spore lengths here, a fair enough percentage of them are around 20 microns long, and there's one of them It might be this one that's in the sort of the lower left. It's like 24 microns long. That's very long for Mitrula elegans. And if you read up on Mitrula, you find out that Mitrula paludosa has long spores, up to 24 microns long. I did not sample this for sequencing because I just thought, well, it's just Mitrula elegans. It's not that interesting. But then I looked up the spore dimensions. So anyway, I got two of these lying on slides right now from a different location. One of them is very much yellow, and the other one is kind of orangey. I want to see if there's any difference there. This one's pretty yellow here. So I want to see if I get the really long spores. I want to see if I have more than one species. But that's stuff that I just found today.
Nice.
So this was from last Saturday. Yeah, it's a reasonably good photo.
And Dave, yeah, cool. 24 is pretty big.
Yeah, really.
But Myco, Quebec, I was just there. Yeah, I mean 11 to 18. 11's pretty short.
You know what? This might be one of these things where these spore measurements just get passed on
Yeah,
from
maybe.
resource to resource for years and years and years. Somebody may have measured them when they were still in the ascus. I don't know.
Yeah.
I mean, it might just be that that's not right,
Sure. Yeah. But Mitrula is always nice to see.
Yeah, well, maybe I'll see what I come up with these ones I have out on slides now.
Oh, I saw this.
So, yeah, some kind of Xylaria in my yard, in leaf litter, growing from a stick that was buried in leaf litter. I don't have any micro on these yet, but these are immature. They're covered in conidia, so I imagine I'm only going to find, just these like variably shaped round conidia. It's probably what I'll find. That's kind of weird. This one has this like nipple. Looks like a baby bottle or something, on the end. That's kind of strange. I put the stick back in.
Growing up again, up through something,
Yeah.
A seed or something.
Oh, it's something got attached to it, maybe.
Or it just was growing up and pushed through whatever it was up against and created this kind of band of. I was stuck here for a second, and then I burst through.
Yeah, that makes sense. So I put this back in my yard in some leaf litter. I put the stick back there. I'll see if they mature, and well, I'm still going to see if anything interesting falls on the microscope slide. I kind of doubt it because they're immature. But I did put the stick back in my yard, and I'll check in a couple of days and see if these things turn black and maybe get some mature spores.
And Dave, it was definitely a stick. One of these photos, I saw it on iNaturalist. Maybe it's the fourth one in the set. I looked at that and I was like, "Oh, is that a magnolia cone?" But yeah, it's not. It's a stick, right?
No, yeah, we don't have any magnolia here. Not that I know of. No, it's on a stick. The stick was buried in damp leaf litter. I cleaned the leaf litter off of these. There were like old rotting leaves stuck to them, to the the dark parts.
Cool. Yeah, that's a cool one.
Yeah, that was pretty interesting. I thought, hopefully I can see it again, when it's more mature, because I know where I put it in my yard. I also found more Melissa in that. This was in the same area where the Melissa is. I found more of those. I put another observation on MO, and I tried sampling the Melissa again. I think I actually got one, like with a razor blade, got one fruit body off of the bark and into one of the DNA wells.
Dave, a good tip that I got from someone in the club that took a bryophyte identification class.
Uh-huh.
Investing like $20 to $30 in a really good set of forceps tweezers
Yeah, I
helps
know.
helps a lot.
Yeah, I imagine. Yep. Yeah, I should think about doing that.
Okay, thank you, Dave. I think that's
Yeah, you're
it.
welcome. Wasn't a terribly exciting week, but yeah, I found a few things. I got more stuff today. Small, mostly small. Oh, I did find Boletus miniata olivaceus today, but I haven't made the observation yet. I want to edit my photos and try to make a good observation. And I found a Lexinum today, also a gray Lexinum, one of the section scabra ones. But maybe
Well,
next week I'll show those.
yeah. Are you expecting the two rainstorms that we're expecting in New Jersey?
Showers off and on
Okay.
through most of Friday, starting tonight. Yeah, it's been dry here. We had 7/10 inches rain on Sunday. But it didn't seem to bring out a whole. I mean, it brought out some things. I found those two boletes. They probably would not have come out if we didn't get that rain. And I found some Lachnum today. I found some Inacebaceae little thing, probably Melosabe. So there are some mycorrhizal species. Oh, I found an Amanita today, a very strange looking version of what I think is Flavoconia, but a really strange one. I think it fruited and then dried out, and so it sort of looks not typical. But I don't have any of that posted yet. I haven't had time to go through my photos.
I found my first Chrysoblema locally too, which is nice.
Oh, okay. Oh, wow, yeah, that's kind of early for that. But I guess not.
Yeah, not
That one
really.
can be early. Yeah, not really. Yep.
Well, I mean, I only find them in this like one area, but yeah, locally. Um, but yeah, I think this is about when they come. It's been so, I mean, it's sort of early, everything here this year. It's so hot.
Uh-huh. Yeah, it's getting warm now. Um, it's supposed to be in the night. It's supposed to be 90 Thursday and Friday
Yeah.
with showers. So I'm sure the amanitas and boletes and russulas and so forth, they're all going to start. They're all going to get going.
Yay.
Hey, is any other suggestions to look at anything tonight, or we'll just wait until next week to get some good observations in? I don't really see anything unnaturalist from other people finding in Jersey. So, Jason, these are all lichens. All right, well, it's only 8:15.
I can share something I saw from Montreal.
Oh, good.
Oh yeah, go for it.
Yay.
Yeah,
Let
we'll
me
take foreign.
let me see how to share from my cell phone.
Where
Share
was
screen, right?
right?
I was at Montreal last week for my daughter's graduation. I'm still trying to Do you see my screen?
Not yet. Now we're coming.
Okay. So Mount Royal is right by McGill University. So every morning I take a walk. I saw some mushroom. We should not see in the summer. So I thought I would just share this. And it's still trying to,
Load. Yeah.
yeah.
You were in Montreal, is that what you said?
Yeah.
Oh, okay, nice.
Uh,
There was probably
I saw mushroom should appear in winter. I don't know why it takes so long. Do you see my screen though?
Yes.
yes.
Okay. So right now I'm showing a gigantic Morel I saw.
Wonderful.
It's bigger than my hand,
Oh boy,
but the only one.
huge. That's huge.
And it's kind of old and brittle, so it broken. But still, if you look, it's definitely a Morel. And I brought back and sauteed it. Doesn't really taste well. It's too old.
Oh, it didn't taste good, or it did?
I mean, it tastes normal, like a normal
Good.
mushroom. But I guess I never had a Morel. I had one or two. I cannot remember how it tastes.
Yeah. You have to make sure you salt them when you cook them.
What do I do?
Salt. Make sure you salt them. Salt the morel.
Okay.
Yeah. The flavor is hidden until you add salt.
I see.
Yeah, this one looks like it's not decayed.
Not decayed. It's just brittle.
Yeah.
Yeah, it looks good.
Okay, so that's Morel. Share another one.
What Morchella americana probably. What did Inat tell us
Okay.
something?
What Inat gave me is
Americana.
Americana.
Okay.
Yeah.
Yeah.
So another one. This one definitely. I saw it. I said, okay, I know what it is. I flip over. It is right.
Oh yeah.
And this should not be in the summer, right?
Spring. They come out in spring.
Oh, they do come up.
It's a
in
little bit
spring.
late though, but
Yeah.
may have been chilly. This was up in Montreal.
Yes.
Yeah, it may have been. They may have had some chilly nights up there.
Yeah. So this is the two. I think it's really out of season. I do see a lot of this, but I saw this in New Jersey also.
Yeah, I find those flammulina some years when I'm looking for morels, I find them. But
Okay.
that would be in early May. Yeah, it's a little bit later. I've never found them this late.
Yeah. So this is the one I saw everywhere. I think in New Jersey and in Montreal. Basically, I think because Montreal, the summer season is short. It's basically everything come out at the same time.
Right.
Yeah, that's basically what I say. There are some other small mushroom like a agaric. I did not take picture. I took three. I thought this is interesting. It's really should be winter, spring, summer. So I have three seasons mushroom.
That's pretty specimen. Yeah. The scales are really nice on that one.
And it does look like a feather speck, right? Okay, let's see if I have anything.
Yeah.
Oh, that's another one.
Or hawk's wings.
Yeah. So this is what AI gave me. I believe it's correct, right? Candlomyces.
Candelianus. Let's
Yeah.
see the under. Let's see an underside.
Okay.
Yeah,
So
maybe.
or may not.
I would just call it Satharella sensu lato. Satharella in the broad sense. It might be Candellomyces.
Okay.
Candellomyces is really, really common. So INET sees a lot of them.
Yeah.
So this INET is user generated AI driven. So because it sees so many Candellomyces, Candelianus, I think it's likely that INET might think that a lot of Satharellas are that. Because
Sapphirellus.
that's its frame of reference, is seeing so many.
Yeah. So this third picture just gave you the different stage, the tiny one.
Yeah, I think this is some other kind of Satharella,
Yeah,
but
Dave,
maybe not. Could be.
Wouldn't you say that cap is pretty dark for
Yeah,
Candolianus?
it's pretty dark.
Candellomyces, yeah.
Yeah, this just looks overall kind of different. The stem
Yeah,
is
it doesn't.
really, really long.
Yeah, and it doesn't have those marginal.
Yeah. It doesn't have the appendiculate stuff,
Yeah.
and the stuff clinging to the near the margin of the cap. I think this is something else, but it could be. It could be Candellomyces, Candelianus, but my guess is it's something else.
Okay.
Some other Satharella. So Candellomyces is a genus that is a split off of Satharella. So it used to be part of Satharella.
Okay.
Hey, did you see that in that last photo, the little veil on the tiny one?
Yeah. It looks like the veil's fairly persistent. It looks like it's trying to form a ring. So Candellomyces has a partial veil. It's not very persistent, but sometimes it does persist. Sometimes it does form a ring. I believe I have seen that in the past. But I think what Brandon said is maybe the most important thing, that these are really pretty dark brown. Candellomyces candelianus is like honey yellow. It fades to almost white. Usually there's a yellow disc, so that sort of the yellow color in the middle of the cap kind of persists even after it fades. These seem kind of dark for that species.
It does seem to have something yellow in the middle.
Yeah. This one right here, this one on the sort of the right center.
Yeah.
That looks a lot like candle, but a lot of the Satharellas fade. A lot of them fade. Most Satharella species are hygrophanous. They change color as they lose moisture.
Mm-hmm.
And then, Dave, in this photo, we can see something that you mentioned that I think points away from Candellomyces as well. That stem to cap ratio would be abnormal,
It's
right?
not right for Candellomyces.
Stature is different. Yeah.
Yeah.
Such a small cap, such a long stem. These are growing from what? Leaf litter?
This is a leaf litter, yes.
Yeah. Candellomyces candelianus is more of a wood border, grassy area kind of species.
Yeah,
Like a
but if you
waste
look at the
area.
tiny one, it is attached to a piece of wood.
Yeah, well, they can do that.
Yeah.
But yeah, I think it's something besides Candellomyces.
Okay, Dave, can you type in the other names?
Oh, the genus. Yeah.
Yeah.
So it's going to be genus Satharella.
Yeah. Yeah, the color
Or
looks different.
or it could be, I'm typing in a few different things. Igor, are you still there? It's sensu lato, right? Lato.
Yes, sensovato.
Yeah. Okay. So I just put something in the so genus Satharella would mean that it's a mushroom in a species that strictly belongs to the genus Satharella. Satharella sensu lato means that it's in a genus that may have been part of Satharella at one time. There's a few other genera. There's Homophron. I don't think this is Homophron, but that's another genus that used to be in part of Satharella. There's a few others as well. I'm just not thinking of them now.
Okay, thank you. I'll stop
Sure.
sharing here.
Okay, well that was good. Those were interesting observations.
Yeah, really nice photos too.
Okay, anybody else have anything they might want to share, get a second opinion on? Okay. Nothing else, we'll call it a night. Thank you, everyone.
All right, thank you.